Hi, this is Ritik, your biology subject matter expert from memoneet. In the previous video, we have discussed regarding the biotechnology and its processes, how where all in the field we normally use this applications and all. So today we will discuss in detail about that. That is the three critical research areas of biotechnology. So very first critical area where the scientists or the researchers focus in the field of biotechnology is providing the best catalyst in the form of improved organism. Usually it could be a microbe or a pure enzyme. The second one is creating an optimal condition through an engineering for a catalyst to act. And the third one is the downstream process technologies to purify the protein or organic compound. So, where all the biotechnology has been applied? Coming to its application in agriculture, it has been used in agrochemical based agriculture, organic agriculture, and genetically engineered crop agriculture. So, these are the three main focused area where it has been applied. So, there comes another topic called green revolution. So, how many of you are very much aware about what do you mean by green revolution? One thing is in during the green revolution the a successful or succeeded in the tripling the food supply, but yet it was not enough to feed the growing human population. So there was a certain time when with the growing human population we were unable to supply a proper food. So that time an increased yields have partially been used to the use of improved crop varieties, but mainly due to the use of the better management, particularly and the practicals, managemental practicals, and the use of agrochemicals such as fertilizers and pesticides. So, for farmers in the developing world, agrochemicals are often too expensive, right? So, the further increase in yield with existing varieties are now possible using a conventional breeding. So that conventional breeding, like, see, if you have a old part of a plant that can be regenerated, say like you have a old, plants could be regenerated from explants. That is any part of the plant taken out and grown in a test tube under a sterile condition when a sufficient nutrient under the media. So these capacity to regenerate a whole plant from any cell. So it is actually called as totipotency. So nutrient media, it must consist of provides a carbon source media which should provide a carbon sources such as sucrose or inorganic salts, vitamins, amino acids, then growth regulatory factors such as cytokines and many more. So, by application of these method, it is possible to achieve the propagation of large number of plants in a very shorter duration. And this method of producing thousands of plants through tissue culture is actually called as micro propagation. So, each of these plants will be genetically identified to the origin plant from which they were grown. That is, they are soma clones. Okay, so many important food plants like tomato, banana, apple, and all they have been produced on commercialized large scale using this method. So another important application of this method is the recovery of healthy plants from a diseased plant. So if a plant is infected with the virus, the meristem, okay, that is the apical and the axillary, which is actually a free of virus, right? Because it is the growing stage of the cell. So that from that you will take the cell, and scientists have been isolated a single cell from a plant, and after digesting the cell wall, they have been also been able to isolate a naked protoplast, so which is surrounded by the plasma membrane. So isolated protoplast from two different varieties of plants, each having a desirable character, can be fused to get the hybrid protoplast, which can be further grown to form a new plant. So these hybrids are called as somatic hybrids, while the process is actually called as somatic hybridization. Just remember. So plants, bacteria, fungi, and animals whose genes have been altered by manipulation, they are actually called as genetically modified organisms. Okay, GMO. So plants have been useful in many ways, and genetically modification has made crops more tolerant to abiotic stress such as cold, drought and salt and heat. So where reduced reliance on chemical pesticides, they don't depend on pest resistant crop. Like uh, they are automatically a pest resistant crop, so they don't require any chemicals, pesticides. Help to reduce post harvest loss and increased efficiency of mineral usage by plants. So this prevents early exaggeration of the fertility of the soil. Enhanced nutritional value of food, that is golden rice, vitamin A enriched rice. In addition to this use, this what a genetically modification has been used to create a tailor made plant to supply alternative sources to industry in the form of starches, fuels, as well as pharmaceuticals companies. So we have BT cotton, which is again a genetically modified plant. Some strain of Bacillus thuringiensis, which produces a protein like kill certain they normally these proteins kill certain insects such as tobacco budworm or army worm, lepidopterans. So then we have beetles, which is actually called as coelopterans and dipterans, like flies, mosquitoes, and all. So this Bacillus thuringiensis forms a protein crystal during a particular phase of their growth. So these crystal contains a toxic uh, Okay, so these toxic, which is called as insercidal proteins. So what happens actually is this BT toxin proteins exist as inactive protoxins, but once they get entered into they are being ingested by an insect, and once they get into the gut of the insect, they become this inactive toxins. They convert into an active form of the toxin due to the alkaline pH in the gut region which solubilize the crystals. So then this activated toxin will bind to the surface of the midgut epithelial cells and create a pores that causes a cell swelling and lysis and eventually causes death of an insect. So specific Bt toxin genes were isolated from the Bacillus thuringiensis and incorporated into the several crop plants such as cotton. So the choice of genes depends upon the crop and the target pest, as most Bt toxins are insect group specific. So the toxin is coded by the gene which is called as CRY-IAC and which is named as CRY. Then the protein encoding by the gene CRY-IAC and cry 2AB. So they normally control the cotton ball ones that are cry 1AB controls the corn borer. So we have two genes here. One is cry 1AB and cry 2AB. So here what happens is this gene that we are talking, right? So they normally controls the cotton that is where it is. Sorry. Yeah, here it is. So basically, what happens is it is gonna control. So this, what you're seeing, will be control the cotton ball worms, and this gene that you're seeing is will control the corn borer. So this is one which has been destroyed by the ball worms, and what you're seeing here is fully matured cotton ball. Next, we have a pest resistance plant. So several nematodes parasites are wide variety of plants and animals, including the human beings. So nematode, namely, they normally infects the roots of the tobacco plants and causes a great what do you say that reduction in the yield. So a novel strategy was been adopted to prevent this infection, which was based on the process called as RNA interference. So where the RNA interference take place in all eukaryotic organisms, which is as a method of the cellular defense that we know. So this method involves silencing of specific mRNA due to the complementary of uh, you know uh, dsRNA molecules that normally binds to, and it also prevents the translation of this mRNA, which is through slicing. So the source of this complementary RNA could be from an infection by virus having RNA genome or mobile genetic elements that replicates an RNA intermediate. So using the agarobacterium vector, so nematode specific genes were introduced into the host cell, and the introduction of the DNA was such that they produced both sense and antisense RNA in the host cells. So these two RNA, what they do is they being complementary to each other, form a double-stranded, that is dsRNA is nothing but double-stranded RNA that initiates the RNA interference and thus silence the specific mRNA of the nematode. So the consequences was that the parasite could not survive in the transgenic host, uh, expressing specific interfering RNA, and the transgenic plant therefore got itself protected from the parasite. So then we have biotechnology's applications in medicine. So in what way they have been applicable in medicine? So like insulin, right? Insulin it have been used for the diabetics was very early extracted from the pancreas of the surgery cattle as well as pig. So what actually happens here is the insulin from the animal sources that we have been extracted, right? So it causes some allergic to certain patient and a type of allergic reaction because they will be considered as a foreign protein. So insulin isolated from two short polypeptide chain. So one is chain A and chain B that are linked together by disulfide bridge here. So the blue color that you are seeing is chain A, and the green color that you are seeing is chain B, and this has been disulfide bridge. So in mammals, what happens, including humans, insulin is synthesized as a prohormone, like a proenzyme. The prohormone also needs to be processed before it becomes fully matured and functional, right? Hormone. So which contains an extra stretched, which is actually called as C peptide, C peptide here. So this C peptide is not present in the matured insulin and is removed during the maturation into the insulin. So the main challenges for production of insulin using the rDNA technology was getting insulin which is resemble into the matured form. So in 1983, Eli Lilly, an American company that prepared two DNA sequences corresponding to A and B chains of human insulin and introduced them in the plasmids of the E. coli to produce the insulin chain. So chain A and B were produced separately, extracted and combined by creating the disulfide bond to form a human insulin. Then we have gene therapy. So gene therapy is collection of the method that allows correction of gene defects that have been diagnosed in the child or during the embryo. So here gene are inserted into the person's cells and tissue to treat the disease. Correction of the genetic defense involves delivery of normal gene into the individual or embryo to take over the function, and compensation of non-functional gene. So the first clinical gene therapy was actually given in 1990 to a 4 years old girl with adolescence dimensions, that is ADA deficiency. So these enzymes is crucial for the immune system to function. The disorder is caused due to the deletion of gene for adolescence dimensions. So in some children, this dimensions that I've been talking, right, deficiency can be cured by bone marrow transplantation, and other it can be treated by enzyme replacement therapy, in which the functional ADA is given to the patient by injection. But the problem with both of these approach that they are not completely curable. Okay. Then lymphocytes from the blood of the patients are grown in the culture outside the body, and the functional ADA, that is cDNA, using a retroviral vector is then introduced into these lymphocytes. so which are subsequently returned to the patient. I'm talking about how do they cure through the genetics. So through the gene insertion. Okay. So as these cells are not immortal, so the patient requires periodic infusion of such genetically engineered lymphocytes. So if the gene isolated from the uh, that is marrow cells producing ADA is introduced into the cell at early embryo stage, it could be permanent cure. So because that embryo while developing itself will get to know like what have to be produced, and that is what it is gonna develop. So the next we have is molecular diagnosis. So where early diagnosis of any diseases and understanding its pathophysiology is very important. Using conventional methods such as diagnosis, serum and urine analysis, and using these things, early detection is not possible. So what we normally do is we go with recombinant DNA technology or PCR, that is polymerase chain reaction or enzyme linked immunoassay, are some of the technique that serves the purpose of early detection. So presence of pathogens such as bacteria and virus and many is normally suspected only when the pathogen's growth is more and it has start producing a disease symptoms. However, at their very low concentration of bacteria or virus, so symptoms you may not be visible, but it can be detected very earlier by using the PCR amplification of the nucleic acid by PCR. So PCR is now rooted used to even detect the HIV in a suspended AIDS patient, so it is being used to detect the mutation in the gene also, if us and suspected cancer patient too. So a single stranded DNA or RNA which is targeted with the radioactive molecule, that is the prop, is allowed to hybridize to its complementary DNA in a clone of the cell, followed by detecting using the radiography. So if the clone having the mutated gene, so that will not appear on the photographic film because the probe, will not be having the complementary gene. So mutated gene will be different. So in that way, we will able to understand that there is a mutation might have happened. So ELISA is based on the principle of antigen antibody, their interaction. So based on that also detection is possible. Next, we have a transgenic animal that have been their DNA manipulation to process and express an extra foreign gene. They are actually called as transgenic animal. So here, transgenic animals such as transgenic rat, rabbits, pig, sheep, cow, fish have been produced. Although over 95% of all these existing transgenic animals are mice, that what we normally use in experiment also. So normal physiology as well as development coming to the transgenic animal can be specifically designed to allow the study of how genes are regulated and how they affect the normal function of the body and its development. Example, study of complex factors involving the growth such as insulin like growth factors. Then by introducing the gene from one specific species that alters the formation of these factors and study their biological effect, that results information is obtained about the biological role of the factor of the body. Then to study certain diseases. Many transgenic animals have been designed to uh, study or to understand how gene contribute to development of the disease and the specificity made to serve as model for human disease so that the investigation of the new treatment for the disease is made possible. And today transgenic models exist for many human diseases such as like cancer we have, and cystic fibrosis, then we have arthritis and Alzheimer's diseases. For that also we normally use this transgenic animal model. So biological products, medicines required to treat certain, human diseases can contain biological products, but some products are often expensive to make. So transgenic animals that produce useful biological products can be created by introduction of the portion of the DNA which codes for a particular product, such as human protein, that is alpha 1 antitrypsin, used to treat the ampicillin. And similar attempts are also been made to treat that is the phenylketonuria as well as the cystic fibrosis. So in 1997, the first transgenic cow, Rossi, produced the human, protein that is which is enriched milk 2.5 grams per liter. That is what they say. The milk contains the human alpha lactoalbumin and was nutritionally more balanced product for human babies than natural cow milk. So then we have is vaccine safety. So here transgenic mice are being developed for use in testing the safety of vaccine also before they have been used in humans. So transgenic mice have been used to test the safety of this polio vaccine. So if successful and found to be reliable, so they can be replaced using the monkey to test the safety of the batches of the vaccine. Chemical safety testing is also being used, like to check its toxicity safety testing. So the product is the same as that used for testing toxicity of the drug. So the transgenic animals are made that carry genes which make them more sensitive to toxic substances than non-transgenic animals. They are then exposed to the toxic substances and the effect studies. So toxicity tests such animals will allow us to obtain result in less time. So we have ethical issues. Uh, Ethical issues is very important. So the Indian government has set up an organization such as GEAC, Genetic Engineering Approved Committee, which will make a decision regarding the validity of the genetic engineering research and the safety of introducing these organisms for public services. So before testing randomly, we cannot test on these even genetically modified animals. So that for that, that becomes an ethical issue. So there is a separate committee. So the modified usage of living organisms for public services, it could be for food or medical medicine sources, have also been created problems with patients granted for the same. So there is growing public anger that certain companies are being granted patents for products and technologies that make use of the genetic materials, plants, and other biological sources that have long been identified, developed, and used by farmers. And they are being locally available in Indian people for specific region and country. So, for example, we have rice, which is a very important food grain that preserves of which goes back thousands. Like it has its own history, thousands of years in Asian agriculture history. There are an estimation over like 2 lakhs varieties of rice in India alone. Okay. So the diversity of rice in India is one of the rich in the world. Basmati rice is distinct for its unique aroma as well as flavor, and 27 documentary varieties of basmati are being grown in India. So there is a reference to basmati in ancient text also, and poetry, folk history as well as been growing for centuries. Whereas in 1997, an American company got patent rights on the basmati rice through the US Patent and Trademark Office. So this allowed the company to sell a new variety of basmati in the US and abroad. So this new variety of basmati had actually been derived from the Indian farmers' varieties. So Indian basmati was crossed with semi-draft varieties and claimed as an invention of a novelty. Then the patent extended for functionally equivalents and implied that other people selling basmati rice could be restricted by the patent. Several attempts have also been made to patent users, products, and processes based on industrial traditional herb medicines, etc. For example, turmeric, neem. So, if we are not vigilant and we do not immediately counter these patent applications, other countries' individual may encase on our rich legacy, and we may not be able to use any of the things that we have already. So, biopiracy is a term that refers to the use of the bioresources by many companies and other organization without proper authorization from the country and people concerned with that compensationary payments. So most of the industrial nations are rich financially but poor in biodiversity and traditional knowledge. So, in contrast to the development and underdeveloping world is rich in biodiversity and traditional knowledge, but you there is no proper resources there. Companies. So, traditional knowledge related to bioresources can be exploited to develop modern applications and can also be used to save time, efforts, and expenditures during their commercialization. So there has also been grown realistic of the injustice, growing some of the injustice inadequate compensation and benefit sharing between developing and developed and developing countries. Therefore, some nations are developing laws to prevent such unauthorized exploitation of their bioresources and traditional knowledge. And the Indian Parliament has recently cleared the second amendment of the Indian Patent Bill that takes such issues into the consideration, including patent terms, emergency provisions, and research and development initiative. So this is all about the entire chapter where I have briefed it very simply and very fast. Regarding this, if you have any doubt, you can contact me or reach me through the comment section or through the Telegram channel. So don't forget to like, subscribe, and also share with your friends. Thank you.